Hi, my name is Sebastian Stockmaier. I'm currently a postdoctoral researcher, and today I'm going to be talking about some of the work I did during my dissertation on vampire bats, and we're going to look at individuals, but also across networks. We're all here because we're interested in this intersect of animal behavior and some aspect of pathogen or parasite biology. And I'm very interested in how social behaviors are affected by pathogens or parasites. And today I'm going to be talking about one mechanism that we term passive self-isolation in a recent review. Passive self-isolation is a result of so-called sickness behaviors like lethargy or social disinterest and can reduce contact between the healthy and the infected individual. Now, I think it's important to distinguish passive self-isolation at this point from something we term active self-isolation, like an ant leaving its nest to protect others from becoming sick, where this behavior has evolved for that purpose. Whereas for sickness behaviors, the adaptive value is still highly debated, ranging from potential indirect benefits such as kin protection to direct benefits like conserving energy for a costly immune response. To induce sickness behaviors in vampire bats, we're using this compound called lipopolysaccharide, which is part of the outer cell membrane of gram-negative bacteria. If you're more interested in the mechanistics behind it, I really recommend reading this recent review. And because we're interested in sickness behaviors in social context, we're studying highly social animals, vampire bats, that have these tractable social behaviors and relationships, and they're fairly easy to work with, so we can bring in an entire roost, we can house them in a fly cage, and it pretty much simulates a social environment they would have in the wild where they spend most of their time in like little tree holes or caves. If you're more interested in social interactions in vampire bats, I recommend checking out this homepage of my collaborator Jerry Carter. So what you can see here is an LPS injected vampire bat, and what we observe in these LPS injected vampire bats is that it reduces grooming given but not received. We're fairly certain at this point that grooming in vampire bats is a low-cost social investment. And what you can see here is a comparison of baseline periods to nights when they were injected with either LPS in yellow versus PBS saline in white. You can see that grooming given almost diminishes to zero, whereas grooming received seems to be fairly unchanged in this setting. We also looked at contact call production. So vampire bats produce these contact calls to attract each other, and you can see a vampire bat that's calling and is soon after being sought out by another individual. And so we injected vampire bats with LPS in yellow or the control in white, and what you can see here are three representative results that just show that contact call production is reduced after LPS injections. And so this could indirectly reduce contact in this species as well. In other studies, we find that grooming between vampire bat mothers and their offspring seems to be less affected by sickness, which is in line with results from other species that show that maternal care can override these sickness behaviors to some extent. We also find that LPS-injected vampire bats receive food donations. So using this experimental system, we don't really see any evidence of avoidance. So having these results now in captivity and from individual bats, we got interested in studying this in the wild and also across entire networks of vampire bats. So I got this amazing opportunity to work with Simon Ripperger, who has developed these proximity sensors that you can glue on the back of individual vampire bats and then track their association events over time in the wild. In this study, we caught a roost of vampire bats and we injected half of them with LPS shown in yellow and half of them with a control shown in white. What you can see here are three different measures of network centrality: degree centrality, which is the number of partners; strength centrality, which is the overall time they spend with others; and eigenvector centrality, which is a measure of influence in the network that quantifies how connected you are to others that are also well connected. And so what you can see here in the treatment period is that for LPS injected bats, those values were always lower compared to the control injected bats. And I think this is important for pathogen transmission, and it shows the potential of this passive self-isolation to impact pathogen transmission. Because if you have a lower number of partners, or you spend less time with others, or you are less influential in a network, that'll affect how a pathogen can spread from you to others throughout a network. So in summary, we find that immune-challenged vampire bats are lethargic, move less, and groom others less, and this effect is lower in close-kin pairs. The main effect seems to be driven by a lower number of social partners, and that's true across individuals and networks, and that's potentially through reduced movement, reduced grooming reciprocation, or reduced contact calling, which is something we show is also diminished. And vampire bats do not seem to avoid their LPS-injected conspecifics in this setting, and even share food with them. With that, I want to say thank you to all my collaborators and all the people that helped throughout various stages of these experiments. And thank you for tuning in.